particular order for the proteobacteria. So we have alpha proteobacteria, beta, gamma, delta. That used to be called epsilon, but now that's campylobacter. All right, so we're going to go with campylobacter. So it's telling you guys that you should be able to differentiate these guys by drawing the dichotomous key. You know how to do that. You know how to draw the dichotomous key. So if you want to do that to make your life simple, you absolutely can, right? But do you want to use all the bacteria for it? Nah. I'll tell you guys which ones are going to be the bacteria we're using. Okay, so when looking at the order for the proteobacteria, this particular grouping is now referred to as the Pseudomonadota. That's the new name given to this particular group, this order. And so this is going to be, these are all gram-negates. They're all chemoheterotrophic. What does that mean? Chemoheterotrophic.
There you go. There.
So they said it louder?
Organic materials
As their carbon and energy source?
Yeah, I think that's what she said.
Yeah. She said it. Yeah. So chemoheterotrophic, a lot of stuff going on, right? So they need their food source from an external source, right? And they get that through chemicals, right? The chemicals is the source, the way in which they get that. So this is the largest taxonomic group of bacteria. You might see that somewhere. So the largest taxonomic group of bacteria, this particular order, and it consists of these 5 classes, right? This is easy. Greek alphabet, alpha, beta, gamma, delta. Easy peasy, right? Campylobacter is the only one that throws it off a little bit, but nonetheless, you have these alpha, beta, gamma, delta proteobacteria, and Campylobacteriota. All right, so let's jump into the alpha proteobacteria. So what is the distinguishing character? Well, before I do that, you guys have that table I gave you? That little characteristics table? So I kind of gave you guys some information, right? It's on the tail end of your PowerPoint. You guys want to talk about?
What table?
You got the package on the tail end of your package. Yep, it's in Canvas too. So I put it in Canvas. I put the student version, I put my table in Canvas too, right? So when talking about the distinguishing characteristics for these guys, alpha proteobacteria, these are capable of growth in nutrient-poor environments. So most of them are capable of growing with very low levels of nutrients, right? So they don't really need a whole lot of nutrients for that growth to take place. They can still grow in low levels of nutrients being available to them. And so when talking about these guys, many of them have stalks or buds or these like extensions known as the prostachia. And so when looking at this particular grouping, there's going to be a couple that I want you guys to know from this particular class. All right, so let's jump into the first one, Pelagibacter. So Pelagibacter is an ocean or aquatic microorganism. So it's one of the most abundant microorganisms in the ocean. It's really, really small, and it typically grows in low nutrient environments, which makes a lot of sense, right? We're talking about the ocean. Because once you start to go down with certain depths of the ocean, you're going to go below, like the oxygen availability is going to decrease. The sunlight is going to be decreasing as well. So you have that photic. Do y'all know about like the layers of the ocean? A little bit, maybe. All right. So when looking at that, like as you go down into the ocean, you have that photic zone, like the zone in which you can see the light. The light's going to be very visible that first 200 meters down, and then once you go past that point, it's going to be aphotic. There's no light. It's going to be dark, right? And so when talking about these organisms though, there's not a lot of nutrients in that environment. When that sunlight starts to diminish and your oxygen is going to start to diminish, right, as you guys gradually go deeper into that ocean waters. And so as a result, these guys can thrive in that environment. They're very important in the carbon cycle, right? So we know how carbon with organic matter, how they're going to be able to cycle through there. These guys are very important for this particular growth. Now, another bacteria that doesn't require a lot of nutrients is Azospirillum. So when looking at this, oh, you know what? So Pelagibacter, way to remember that one that's going to be found in the water. Pelagic actually means open waters. Like that little prefix, Pelagi, that actually means open waters. All right, so Pelagibacter found in open waters, ocean. Because in the environmental sciences world, we have the pelagic zone. The pelagic zone is like the really deep depths of the ocean waters. Yep. So Azospirillum. All right, told you a couple of things about this bad boy. So azo, we're doing something with nitrogen. Whenever you see that prefix azo, something's gonna be going on with nitrogen there. All right, that azo prefix tells us that something's gonna be going on with nitrogen. Spirillum, that's gonna be the shape, right? So Azospirillum, these guys grow in soil and they use nutrients excreted by plants, right? So we know that plants are gonna be making what? When talking about photosynthesis, they're gonna be making sugars and oxygen, right? So we're making sugars and oxygen. And so they're going to be forming and associated with many roots as a result of this, right? That makes sense because if they're forming the association with the roots, that means that the stuff that the plants make, they'll be able to come in contact with it because they're already there by the plants, by the roots. something else is hanging out by fungi groups too. Do you guys know? Nah, I said it. Dang, fungi. I said it. So fungi actually hang out with plant roots as well to kind of get those sugars. They want the sugars because they use the sugars to make cellular respiration take place, right? So they do this with plants such as tropical grasses and sugarcane. So remember I was saying that AZO prefix means something about nitrogen? These guys fix nitrogen. What does that mean? All right, there's this process called nitrogen fixation. All right, nitrogen fixation. And with nitrogen fixation, it's basically when you're converting atmospheric nitrogen to a usable form. All right, so you're converting atmospheric nitrogen, that's atmospheric nitrogen, into a usable form. So that could be nitrite, that could be nitrate, that could be ammonia, that could be ammonium. All right, so nitrogen fixation is when you convert you convert nitrogen, atmospheric nitrogen into a usable form. That could be nitrite, that could be nitrate, that could be ammonia, or that could be ammonium. Nitrogen fixation. So that's great because some things cannot use nitrogen just like that atmospherically. It has to be in some sort of type of form, right? This is great for other organisms, right? The cycle of life, right? Okay. So then we have the Acidobacteriaceae. This is a combination of Acinetobacter and Gluconobacter, and these guys are going to be involved with converting ethanol into acetic acid. What's that? We've learned about that before. So yeah, vinegar is definitely acetic acid, but what's that process called where we're able to convert ethanol into acetic acid or vinegar? Fermentation. Which one of those fermentation processes?
Anabolic.
Well, it's not really anabolic. It's gonna be a catabolic. But what is that process? We have two types of fermentation, right? What are those two types of fermentation?
Where you use like an organic source and non-organic source.
One starts with an A, alcohol fermentation. The other starts with an L,
Lactose.
lactic acid fermentation. There we go. Okay, so these guys are gonna be able to carry out alcohol fermentation, right? Converting the ethanol into acetic acid. So see how everything comes back together? Yeah.
So this one is just alcohol?
Yeah,
Fermentation?
alcohol fermentation.
Okay.
Yep. All right, so in that table though, I think that, so in the table I combined them, right? So in the table I combined the Acinetobacter and the Gluconobacter together, all right? So, and I even put down the AZO, the nitrogen fixing back. That's so nice. All right, so yeah, you guys have some good notes here, right? So we're looking at the Acinetobacter and Conobacter, that's that combination with the process for fermentation to take place, all right? And so we're looking at the next microorganism, okay, Rickettsia. All right, so Rickettsia is interesting because it's an obligate intercellular parasite. What does that mean?
Obligate that it has to
It has to,
do the next thing.
that's to do what?
The next.
The next
Wait,
thing,
what
which
was it?
is? You said it. Yeah, so it has to live inside of a host. You said the next thing, that's the next thing. So it has to live inside of a host, right? So obligate intercellular parasites, is that living?
Yes,
Yes.
because it's still stuff.
It still has a cell, right? It's still a bacterium, it still has a cell, right? So that's one of the, remember I kept telling you guys about viruses, like, do not let the fact that they require host cell, like, trip you up, because we have things that are living that require host cells too. It's the fact that they don't have cells, that's why viruses are non-living. Viruses do not have cells, they're not alive. All right, they just look alive sometimes, but they're not. So Rickettsia are going to be transmitted by insects and tick bites. So we want to be very careful, right? I know that ticks are a thing out here, so be very mindful of that. So these guys are going to be able to cause spotted fevers, such as the epidemic typhus, which is Rickettsia prowazekii, Rickettsia typhi, which is the endemic murine typhus. That's going to be for mice, right? Murine, whenever you see that murine, that means something with mice or a mouse. Right, murine is a mouse, mice, however you want to phrase it, and then Rickettsia rickettsii is rocky mountain spotted fever, which is more common than the epidemic typhus. All right, so when looking at the, figure right here, this is a picture of rickettsia. I went ahead and pulled that from Wikipedia because it shows those little small little structures right there. That's the actual parasite inside of a host cell. All right, that's the parasite inside of a host cell. Just pulled it from Wikipedia. It's a real picture. All right. So one thing I tell people about Wikipedia. All right, Wikipedia is cool if you check the references. Do not use Wikipedia solely. Never, ever solely use Wikipedia. You can use it as a starting point because you have to check those references. And the reason why I know you can check the references is because, like, I get bored sometimes and I go and I add stuff too. That's a hobby. I update Wikipedia pages. I used to do it a lot when I had more time, but I would update Wikipedia pages. And all you gotta do is like make the updates and you put your name on it, and like somebody comes behind and fact check and they might leave it. Now sometimes I do stuff to be funny, and I'll put it in and see how fast they take it out. Like I don't do science stuff. Like I was talking about like how you have like resources, competition for resources between siblings. Like how siblings, like with your siblings, you get along very well as adults, right? But when you were kids, y'all probably like toys because you guys were vying for your mom's attention. But now that you guys don't need their attention anymore, it's separate, right? So that's the type of stuff that I would do. Hold the conversation. But yeah, Wikipedia is valid. Check the sources. You should be good to go. And I'm telling you that for your lab report. All right. So for your lab report, you can use Wikipedia for some things. Just check it. Go to the original source and go off there. All right. Start from there probably, and then go to the original source. All right. Ehrlichia. So Ehrlichia is a type of alpha proteobacterium. And so these are also transmitted by ticks, and this one causes ehrlichiosis. And so I was going to ask you guys, since these guys are going to be part of alpha proteobacteria, what do they have in common then?
Growing with low levels of nutrients.
Yes, that's the key indicator right there. They grow with low levels of nutrients. Okay, so make sure you guys remember that. Like, okay, that's why they're grouped together, because they grow with low levels of nutrients. All right, so this particular one is a human pathogen, and it could be fatal in humans if it's not treated properly for ehrlichiosis. Okay, so that's something to keep in mind. So this particular one is a human pathogen, and unfortunately, this bad boy can be fatal in some instances. And so, also to add for Ehrlichia, this one can target your white blood cells. All right, so I made a note of that on my table, but this particular one is going to target the white blood cells. All right, that's bad, right? Because your white blood cells help you with your immune response, right? So that's something to keep in mind for these two. Well, this one, ehrlichiosis. All right, so looking at this figure here. This one shows a picture, figure 11-1 in your text, of the rickettsia. And so here it shows a chicken embryo cell. So I don't know if you guys ever seen that, but you can actually see the chicken embryo cell development online. Go to YouTube, type it in, you can see how that progresses by the days or whatnot. So this one's just showing a rickettsial cell that has just been released from that particular host cell, has a slime layer around it. What's a slime layer?
Glycoproteins.
So not biofilm, it's similar. It's made of proteins and carbohydrates, because it's going to be a type of, it's associated with the capsules, right? So the capsule's going to have that hard structure for the glycocalyx, right? So it's a glycocalyx. So the capsule is the hard, firm structure. The slime layer is going to be more loose and
It's
not as,
it's sun protection, and it can also help for motility
helps with,
because it less friction if it's on the surface.
it can adhere to surfaces because of that particular slime layer by being kind of loose. It really helps with virulence. With the virulence of that particular microbe, right? Because if you can't access it, you can't get to it, then how can you kill it? All right. So when looking at this one, we see the structure of that particular rickettsia, but then we see like this chicken embryo cell, and you have a lot of these ricketsias, and unfortunately they're in the nucleus of that chicken embryo cell, so they're gonna definitely use that host cell's machinery to make more and more copies of itself, right? That's how you get those masses, is it's using that cell's DNA to make more and more copies of itself, and unfortunately, it's going to destroy that cell, right? It's going to destroy that cell at some point. All right, so these ricketsias are scattered, and you have some masses in the nucleus, so not too good for that embryo. All right, so some more alpha proteobacteria. You have Caulobacter. So Caulobacter is Hypomicrobium. And so these are going to be found in low nutrient aquatic environments, and they have those little stalks, the Prosthepiae on these guys. And so when talking about these structures right here, these guys reproduce through budding. All right, so these guys reproduce through budding rather than binary fission. That's different because we said that bacteria produce through binary fission, right? Where one cell splits to form two. budding is interesting because budding can be asymmetrical. It not can be, budding is gonna be asymmetrical, right? That's when you have a structure that like branches off from that main structure. And when it does so, it doesn't always take an exact copy of information, right? Budding can like just branch off and not take an exact copy of the information. So the one thing I had on my notes for Hyphomicrobacterium I wanted to add was that Hyphomicrobacterium, these are found in laboratory water baths. We have a laboratory water bath, right? So that means you keep that stuff clean, right? Because they're found in low nutrient aquatic environments. So like our water baths, we don't put any nutrients in it, we just put water in it. So that's why when you start to get that film layer, like on a water bath, you gotta clean that.
Where's that come from? Is it in the air and then it
So,
gets to
with-
it or what?
So, great question. So these microbes, they don't necessarily come from the air. No, it has to come from some type of water source. So let's say, for instance, with you working with aquatic samples, these might be tag-alongs. So if it's stored in another type of tube and you transfer it and it's already coming from another source, then it could be present in that environment.
So it could technically even be like in DI water containers?
Ooh, that's not good. If it's in DI water, then no, that's not good because that means your DI water source is contaminated. So if there's contamination in DI water, then yeah, but that should not happen. But yeah, because like that can actually happen with one we're gonna talk about in a little bit, Legionella. Legionella is one that if it gets in your water, your air conditioning vents, you get really sick. It's not that common. It's not freak you out. I'll tell you guys about that though. All right, so Caulobacter, when looking at this particular microbe, this one is going to be showing figure 11-2. And so you have this picture that shows a transmission electron micrograph of these little guys. And so here you see that you have the flagellated swarmer cell. What's swarmer? What does that mean? Swarmer, swarming? They can move, right? They can move. So remember I told you about Proteus last week? Proteus can swarm on the plate. I showed you guys how you have those little lines on your plate for the Proteus. Yeah, they can move too. So these are and that makes sense, they're aquatic. They're found in the water. They have a flagellum too. So these guys are gonna have this flagellum structure loss. And when that flagellum structure loss, you're gonna have a stalk that begins to form and eventually elongates to where you're gonna have the division of that cell to where you now have the two flagellum present to where you're gonna have one per cell. Right, so two flagella, because you're gonna have one flagellum per cell. And so you now have these new cells with their flagellum ready to go out and swarm and make more and more copies of themselves. All right? And so then you have the Hyphomicrobacterium. With this particular one, it has its little bud structure, and then hypha, that should sound, well, not yet. It will sound more familiar in chapter 12. When we start talking about the eukaryotes, because hypha is a structure found in fungi. All right, so that's a little similarity that we see right here. Looks a little bit like a fungal structure. All right, it's a budding bacterium, which makes sense because budding happens in fungi. Right, budding happens in fungi. All right, so now let's talk about some fun stuff. Okay, so I like Rhizobium, Radiorhizobium, and Agrobacterium. So I used to work with Agrobacterium. So when talking about Rhizobium, Radiorhizobium, and Agrobacterium, why are they all together? They're all together because they're plant microbes, right? These are associated with plants. So with the Rhizobium, Rhizobium and Radiorhizobium, they're gonna be working with something with the nitrogen fixation. So remember I talked about how atmospheric nitrogen can be converted into a useful form. So this is cool at end too, but it's not really useful for organisms, not for plants. No, not really. So this atmospheric nitrogen is converted by bacteria into a useful form for our plants to use. All right, for our plants to use. And so they're gonna fix the nitrogen in the roots of the leguminous plants, right? So the legumes, the plants of that nature, that's great for the plants because they're able to now have that resource, right? And so when looking at this particular group of these two bacteria, they're known by the common name of the Rhizobia. All right, they're known by the common name Rhizobia. Now, Agrobacterium though are actually pathogens. They cause disease in plants. Now, I'm pretty sure you've probably seen it before. You just didn't know you were looking at an infected plant. All right, they cause a crown gall disease. All right, so they cause crown gall disease, and what happens is this particular bacterium, its plasmid will be inserted into the plant cells. And what do we know about plasmids? They can alter DNA. Yep, they're extra chromosome, they can alter DNA, and they can do so when they alter the DNA, they can induce a tumor to be present on that bacterium. You guys want to see a plant tumor? Let's see. All right, so with the plant tumor, I've actually seen this in real time. I actually caused this one time on some plants in an experiment in the lab. We wanted to see how long it would it take for tumor development to happen on plants. And so what we did was we would take some little bit of the agrobacterium, we put it on that plant, the plant seed, and just watch it do its thing, or we put on the branches too. We put on the branch to see, because we wanted to form on the branch, we put it on the, somewhere in the stem. And so here, this particular infection is taking place on the stem of that plant. And so the problem with crown gall disease is that this is blocking the plants from taking up excess nutrients, right? Because you're blocking the stem in itself, but it's probably gonna be blocking some of that root formation below that, and the water absorption is gonna be affected. The plant's not gonna be able to take up nutrients like it wants to, and that plant's gonna eventually, unfortunately, shrivel up and, die. Unless that tumor can move.
So is it something specific about that particular plasmid in that particular bacteria that causes tumor, or can any plasmid cause
Great
changes?
question. It's
Like
that
if you
specific.
physically inject it or something?
Yeah, it's that specific plant, right? So that specific, not that specific, that specific, bacteria. So agrobacterium in itself is a plant pathogen. If we were to use rhizobium or bradyrhizobium, what's causing it is not a plant pathogen, right? So those two, rhizobium, bradyrhizobium, it was another plant pathogen too that I just mentioned a little while ago. It was azospirillum. It's not a plant pathogen, it's a plant microbe. Plant microbe. Those are all plant microbes, right? But this is the actual pathogen. This is cause disease. It's going to known to cause disease. If you want to look it up for more, it's called Agrobacterium tumefaciens. That's the one that's causing it. So tumefaciens is tumefaciens. Tumefaciens. Agrobacterium tumefaciens cause that. I used to work with that one in the lab. It's pretty cool. It's BSL-1. It's not even BSL-2. It's BSL-1. Because it's gonna not affect us, right? It's a plant pathogen, not a human pathogen. Yeah, it doesn't affect human cells, so. Alright. So, with that being said, we have the next one, Bartonella. So Bartonella actually is a human pathogen. This particular one, Bartonella henselaces, cat scratch disease. You also have Brucella. So Brucella is an obligate parasite of mammals. So that means it's required, right, to grow within the mammal cells, mammalian cells. And it survives phagocytosis. What does that mean? It doesn't get eaten up. It can be inside of cells, right? It's gonna not be destroyed. So what's really interesting about this one is that when you think about it, okay, it's an obligate parasite of mammals, so that means that it can thrive inside of mammalian cells. It can survive phagocytosis. So I'm guessing this bad boy can survive within the phagolysosome. Did I go too far? Phagolysosome, does that ring a bell? That's normally what digests those different pathogens, right? So the phagolysosome normally digests or break down these different pathogens, but like, these guys don't have to happen, all right? So this one causes brucellosis. There's another type of pathogen for you guys to remember, all right? So that's gonna be something that's very interesting. All right, so a couple more guys, a couple more. Okay, so we have Nitrobacter and Nitrosomus, all right? So Nitrobacter and Nitrosomus. And so when talking about these two, these are gonna be very important as well for nitrogen fixation, right? So looking at these guys, these are chemoautotrophic. What does that mean?
What she said earlier.
It's a little bit different than what she said earlier. So close. It makes auto is where, that's where it made it a little bit different, right? So auto means self, right? So it makes its own food. It's gonna be using chemicals though as its energy source. It makes its own food, uses chemicals as its energy source. So what are these chemicals? They're inorganic chemicals, all right? They're inorganic chemicals. And so the carbon dioxide will be its carbon source though, because it's gonna make its own food. And when talking about nitrosomas, they're gonna be doing different ways of converting the ammonium product. So like this is ammonium. So ammonium converted into nitrite. And then for nitrobacter, we'll convert nitrite into nitrate, all right? So when looking at these examples, they have different ways in which they're gonna be converting nitrogen into useful forms. And so when looking at these processes, they're gonna be referring to these processes as nitrification. So nitrification, the conversion of these nitrogen products into other compounds, nitrogen compounds, all right? All right, so one of my favorite, I learned about this one a couple years back, it's pretty cool. So this one is called Wolbachia. So this is really interesting because this one lives inside of insects. Endosymbiont, we learned that word. Endosymbiont means that something can live harmoniously inside of something else. Well, this isn't really harmonious. These guys will live inside of an insect, but they will cause problems. They're gonna affect the reproduction of those insects. They can actually kill those insects from the inside. They can hang tight, they can lie latent, hang tight, and then they can actually make copies of themselves and then affect that insect's ability to reproduce. And so that's a good way to have insect control take place, right? As long as it's not a human pathogen, it doesn't affect us, right? So that's a really good way to keep insect control together, if you're able to find those bacteria that can thrive in the guts of those insects, all right? And so what it actually does is affect the reproduction of insects by the male to female ratios and fertility for these guys. So it affects the ratios of male to females and fertility for these guys. Alrighty? Okay, so that was a lot, and that was fun. So what bacteria do I want you guys to know, right? Okay, so when looking at these guys, I would like for you guys to know, of course, your, always the ones that cause human disease, right? So Rickettsia, Ehrlichia, those definitely, because they cause human disease. Your Bartonella, Brucella, most definitely. And throw your Wolbachia in there, all right? The gut insects, affecting gut insects, all right? So the other ones, they're gonna be referred to as our plant pathogens, all right? So do I want you guys to know a lot of details about them? No. But I do want you to know that like Azospirillum is gonna be something with nitrogen conversion, Rhizobium nitrogen conversion, your Brady Rhizobium, your Nitrobacter nitrosomas, things like that. That's very basic sublevel for those, for the plants. But for the human causes pathogens, I do want you guys to know the information about them that's given to you in the textbook PowerPoint, essentially, in the PowerPoint, right? So do I want you to go out there and look up what the what Ehrlichia does as far as the genetics goes? No, we're not talking about genetics, right? I do want you to know that, for instance, these guys are all gram what? Negative. They're gram negative, right? And then some ways to differentiate could be the fact that some may be chemoautotrophic, some may be chemoheterotrophic, well, they're all chemoheterotrophic. So the chemoheterotrophic for the human pathogens, right? Also the fact that they can cause different diseases. So this is gonna be transmitted by ticks, right? These are transmitted by ticks for those. So that's another way to differentiate these different types of infections. All right, so just those types of information at that level. Do I want you to go outside the textbook, outside the lab, outside the lecture? No.
So on the student bacteria chart, under the alpha,
I only listed
just Rickettsia,
Rickettsia.
but we should focus on the ones you just
The
said
one
too
that I mentioned,
also.
yeah. So in my what's that thing called? My version of the chart, I have everything laid out. Yep, I have everything laid out for all of them. But I'm not gonna make you guys know all of them. So for right now, the takeaway for today, going through the list, I'm gonna make sure I put an asterisk next to it too. So that, and you know what, this is recorded. I'm recording this right now. So we're looking at this particular one, like I mentioned to you guys before, you have Ehrlichia, Rickettsia, I already highlighted mine, Ehrlichia, Rickettsia, Bartonella, Brucella, and Wolbachia. The plants are cool. Just know, you know, when you see those nitrogen fixing ones, you know, nitrogen fixation, that's it, that's all I care about, no other details. All right, so we're down one class of these gram negatives. What's next? What's the next class? Beta. So beta proteobacteria, but first, if you want to, on your own, practice making dichotomous keys, you can do that, because on your study guide, I do ask that you guys be able to interpret a dichotomous key. So I made you create one, right, in lab. That's why I had you guys do that. Well, you had to do it for the unknown report, but you also need to do it for here. So if you want to make your own key doing this, you can. Just show it to me. I'll look at it. All right? So for the beta proteobacteria, what are the characteristics for these guys? So these guys are gram what? Negative. Everything's gram negative. So these guys are gram negative. And so when looking at these, very interesting, very different too. So for the first one, the Spirillum, this particular microbe, this one's gonna be an aerobic microbe. All right, so this particular one is gonna be an aerobic microbe, and it's gonna be able to be found in fresh water, and it moves with a polar flagella. What does that mean? Polar flagella. So polar means that it has, what was it, Zingland League?
On either side.
Well, with polar flagellum, it can be one flagellum right there, right? But it's on the poles, on the ends of that cell, right? So flagellum on the ends of that cell. All right, so the Sphaerotillus, this one's gonna be found in the sewage and our freshwater, so aquatic. So these are so-called aquatic microbes, right? And so when looking at these guys, they're gonna form sheaths. What is that? A covering surrounding the structure of these particular bacteria. And so they form a covering or a sheath around them for protection, as well as for them to gather more nutrients, right? So a way for them to be protected and to gather more nutrients. Cool. So looking at these Spirillum volutans, this shows the spiral-shaped bacterium with its flagella on opposite ends of that particular cell. So this one has polar flagella, and they're on both ends of that particular cell. And when looking at that one, you have a lot of them in one space too, right? So we have a lot of them in one space, so it's gonna be called like tufts of the bacteria in one space, but they're polar nonetheless. Alright? Alright, so the Spiracillus natans, you see that you have the bacterial cells, and the bacterial cells are present, and there can be a covering surrounding those bacterial cells, so that way it can absorb or take in more nutrients as it needs to do so. Alright? Alright, so this is where I wanted to get to. Burkholderia, yes. So we're talking about Burkholderia. This is also a gram-negative microbe. This little fella is going to be aerobic. And this one is interesting because Burkholderia sapientia can actually degrade more than 100 organic molecules. What does that even mean? That's not good. Doesn't sound so good. Alright, what about disinfectants that they use in hospital settings? What are those made up of? Chemicals. And many of those chemicals are, so we have Cavicide. I've used Cavicide before. We have Sanisol, Cavicide. There's a lot of different disinfectants, but the thing about those disinfectants, you have quaternary ammonium, all that. The things about those disinfectants is that they contain organic molecules. All right, so this particular one can actually contaminate hospital disinfectants or disinfectants in general. That's why I told you guys that if your disinfectants get cloudy, throw them away.
We found one after, at somewhere we were working at, so I was really glad you told us about that.
Yeah. Whenever I was telling somebody at the nail salon one time, like I was back home and they had some hand sanitizer, and I was like, I think you might want to throw that away. It's a big quality. Like I'm not using it, but I think you might want to throw it away too, though. And I explained it to the lady, and her daughter, she was like 11. She came over there, and I was explaining it to her. She was like, "That's so cool." I was like, "Right?" And like me and the little girl, like we're not BFFs. I don't even know that baby like that, but like we're not BFFs. I go see her and all that stuff. And yeah, so with that, throw that stuff away if you see that it's cloudy or contaminated. All right. So when looking at Burkholderia pseudomeli, it causes melanosis. I'm not gonna worry about that one too much. It's that's sapientia. That's the one. Now Bordetella, this one is one you should have some familiarity with. So Bordetella pertussis causes whooping cough, right? We know that that's going to be something you can take a vaccine for, right? That DTaP, that's the DTaP is the tetanus, pertussis. What's the D? Diphtheria.
Is this the one that causes kennel cough in dogs? Is this the one that causes kennel cough in dogs?
I think it's a different one for dogs. Yeah. So for dogs, it's a different one altogether because I know what the one that causes the kennel cough in dogs because I had it's another one. Yep, it's another one, and that particular one, I'm gonna look it up for that one. It's Bordetella bronchiceptica. Yeah, so I'm gonna say I know what you were talking about. I knew it wasn't this one, but yeah, yep. In the same family though. Well, same family, same genus, different species. Yep. All right, so these are non-motile rods. So they're rod-shaped bacteria, and they're not non-motile. They're not moving. All right, they're not moving. So it makes sense when you think about it. If you're getting into your upper respiratory tract, right? They adhere to those different areas in the respiratory system. They're not moving. The cilia lining your respiratory tract. Would have to move them, so they're just hang tight there, right? Makes sense. All right, so we're looking at the next one, Neisseria. So we know about that one. These are cocci-shaped bacteria, right? We looked at them in lab. So these are going to be our gram-negative cocci-shaped bacteria. They're typically diplococces too, right? They're typically diplococces. And so we have Neisseria gonorrhea, which is the causative agent of the STI, gonorrhea. And then we have Neisseria meningitis, which is the causative agent of meningococcal meningitis. Like, whoa, different sides of the coin? Absolutely, different species altogether. All right, so that's why when someone can get meningitis, they typically do like a spinal tap to see whether or not there's the bacterium present in the spinal fluid. Because if there's an indication of bacteria in the spinal fluid, you most likely have meningitis. All right, and that's going to be a series of treatments for that particular one when you're trying to get rid of that. You should not have any fluids there. So this one is a human pathogen, and it's going to live in the mucous membranes of the mammals, right? So it's going to be the mucous membranes, which makes sense, right? So for gonorrhea, in the vaginal lining, the penile lining of the urethra, so that makes sense, mucous membranes. And then for the meningococcal, we're going to talk about the upper respiratory, getting in our throats, nasal cavities, things of that nature. That makes sense. All right, so Zooplea, this is another gram-neg, of course, and it's going to be very important in the activity of the activated sludge system, so sewage treatment. So it's going to be pretty important for sewage treatment. All right. And so here we have some gonorrhea. So when looking at gonorrhea, the gram-negative cocci-shaped bacterium in this particular area interacting with the host cell membrane, it's gonna interact with the host cell membrane. You remember I say diplococci, so you most likely see those guys in pairs. And yeah, they're going to adhere to that surface until they're taken into that cell and cause the infection, all right? So, what are the important Bordetella that I want you, not Bordetella, the important proteobacteria I want you guys to know? So I put a line next to Burkholderia, Bordetella, and Neisseria. Those are the three. Those are our three for today. So Burkholderia, Bordetella, and Neisseria. So what do I want you guys to know? I want you guys to know, like, how can you actually come in contact with those? Like for gonorrhea, how do you get that one? It's an STI. You also have meningitis. What membranes are going to be affected? Well, what portals of entry? So for the STI, of course, that'll be the vaginal penile areas for the method of entry, mucous membranes specifically, mucous membranes of those structures. For the meningitis, we're going to be up here, upper respiratory, mucous membranes, maybe of your nose, your mouth, right? So yeah, those three. All the other ones, they're cool, but like, All right, so make a key that makes you happy. All right, gamma. Okay, gamma is fun because this is the largest class of the proteobacteria, or the Pseudomodota. So this is the largest class, right? These are going to be very important because these are going to be finding our enterics going to be hanging out over here. All right, so these are pretty important. Okay, so the first one, the Acidophilobacillus. So the Acidophilobacillus is going to be important because it's going to be able to oxidize. What does oxidize mean? Transfer of electrons. Okay, so for oxidation, will you like be losing electrons for oxidation or gaining electrons? Losing electrons, right? So we're gonna have an oxidation of hydrogen sulfide, and whenever you see like a chemical symbol and the superscript O, that means it's gonna be like elemental, right? So elemental sulfur. That means I always want to look over. There's in the other class, there's a periodic table right there. I always want to just walk over to it. But elemental sulfur means that we go to the periodic table, bam, sulfur, right? So oxidizes hydrogen sulfide or elemental sulfur into sulfates, all right? And so when looking at the next one, the triticale. So this particular name, I wanted to highlight this one because of the ALES suffix. So whenever you see the ALES suffix, that means it's going to be in order. Right, so it's gonna be an order, and so if it's an order, that's gonna be kingdom, class, order, family, right? So it's gonna be an order, comes above this family of bacteria. So this particular one, Vegetoa, is gonna be important because it grows in aquatic sediments, and it's gonna be able to oxidize the hydrogen sulfide into elemental sulfur for energy purposes. All right, so it grows in aquatic sediments. And so the other thing I wanted to mention was, as a brief aside, this one, let's see. Okay, we don't mention it, that one. But there's this bacterium known as the biggest bacterium today. It's called Thea margarita. So it's called Thea margarita nambiensis. Like, why do I know margarita, right? But it really is. The largest bacterium today is Thea margarita nambiensis. It's pretty cool if you wanted to look it up. I'll write the name out for you guys. It's in this particular group. So it's going to be a gamma proteobacterium. Thea margarita. You can actually look at it on the light microscope that we have, like very easily. Thea margarita nambiensis. Wait, why did I put a TH right there? Thea margarita nambiensis. That's insane. All right, nambiensis. All right, so the next microbe we have here is Francisella. So Francisella tularensis. If I can get it out. Francisella tularensis is a mammal pathogen, so it doesn't really affect us per se, but it's a mammal pathogen. And this one requires special nutrients for it to grow. It's going to be able to grow on blood agar. All right, so this particular one grows on blood agar. So we talked about blood agar, right? In class, what do you guys remember about blood agar? For strep. It's for strep. Look at you, very good memory. So blood agar is for strep because normally we can differentiate the different types of strep on blood agar plates because some can completely destroy red blood cells, some partially can destroy red blood cells, and some not so much. Right? So that's how you can tell how harmful a microbe could be. So tularemia is going to be rabbit fever. All right, so tularemia is going to be rabbit fever. So just to write that off, won't make you guys know that, but if you go off, sounds familiar. You've seen that before. right? We've seen Pseudomonas. So Pseudomonas is very important. All right, Pseudomonas is a very important microbe. So put an asterisk next to this one. It's definitely going to be some good information about Pseudomonas on your exam because it's very important. Okay. So Pseudomonas is common in soil and other natural environments. 2 terms we've learned before. Opportunistic pathogen. What does that mean?
It targets like a weak immune system.
Second.
It targets a weak immune system.
Targets weaken immune systems, right? So if someone's already sick, they're more susceptible to contracting this one. All right. And then it's a nosocomial infection. What does that mean? Hospital-acquired infection. Hospital-acquired infection. That's really bad, right? So if it's opportunistic and it's a hospital-acquired infection, it makes sense though too, because if you're somebody that you got it in the hospital, you're already sick. You're there for a reason, right? You're not there just to be hanging out in the hospital. Like I just want to be here. No, you're sick. That's why you're there. All right. So, unless you work there, right? So these are metabolically diverse as well. So that means that they're able to carry out these different metabolic processes and have different yields of products. And they're also polar flagella. They have the polar flagella on the ends of them, so they're able to move a lot. So one of significance I want you guys to remember. So remember Pseudomonas aeruginosa. Okay, Pseudomonas aeruginosa is problematic because it's going to be a nosocomial infection. It's going to be an opportunistic pathogen. Of course, but this one is known for targeting wound and urinary tract infections, which makes sense, right? Because how would a wound, you would probably be taking care of a wound in a hospital setting, right? In a hospital setting, you'd be treating a wound, taking care of it, so improper care can lead to the transference of that bacterium, because it's already in the hospital anyways, right? Also, urinary tract infections, how can you get that as a UTI? So, a couple ways. Catheters, yeah. So with catheters, if you guys work in a hospital setting, or when you work in a hospital setting, make sure you guys keep those sterile, as clean as possible. Sterile when you first take it out the container, but clean as possible once you have that applied to your patient. So that way you're not potentially making your patient sick. Because remember, with Pseudomonas aeruginosa, it can get on the catheter, and there can be a biofilm, and that can just hang out. All right, and make that person very ill. Okay. So Pseudomonas aeruginosa is a very important one. Also, this one can sometimes have a level of resistance to certain antibiotics too. So that's a problem. All right, this one has a little bit of resistance to antibiotics as well. So it's like, dang, it's just bad all the way around. And so when looking at this one, it can also synthesize many enzymes. That's why it says metabolically diverse, because it can synthesize or make a lot of different types of enzymes to carry out different processes. All right. All right. Now, what's one thing, this is kind of like as an aside, but they actually can fluoresce. So there's one that I've worked with before. It's called Pseudomonas fluorescens. It fluoresces yellow. All right. This one, if you're working with Pseudomonas aeruginosa, you're gonna know it because it fluoresces blue-green. Right, it fluoresces blue-green, which is pretty. It's cool in conversation, just in reality, you just don't want to have it. Right. It's cool in conversation. That's so pretty. So when you look at the Pseudomonas underneath the microscope, you have that polar flagella, you have that rod-shaped bacteria, right? And so you look at this one though, you can see that this one has like these sections as if it almost wants to undergo binary fission. This is a regular shaped Pseudomonas, whereas this one looks like it wants to undergo division, right? Binary fission makes an exact replica of itself because of that partition, those partitions forming a little bit. Alrighty. Alright, so still looking at this particular order of these Pseudomonales, you have the Actobacter, Azomonas. What do we know about that prefix, the AZO prefix? Something about nitrogen fixation is going on here. So yeah, they fix nitrogen, those two. All right, you have the Morazella lacunata, which causes conjunctivitis. All right, you also have the Acinetobacter. So Acinetobacter, this particular microbe, is typically found in pears, and it is a respiratory pathogen that's, unfortunately, when we look at Acinetobacter baumannii, it's going to be on one of the lists for a ones to watch. Because this particular one is just resistant to antibiotics. That's the problem, right? So if it's resistant to antibiotics, we can find other ways to treat it or to kill it. Not treat it, to kill it. All right, so it's resistant to many antibiotics. So it's one of those problematic bacteria that we have to keep an eye on. It's also opportunistic too, unfortunately. So it's also opportunistic. All right, and it's naturally found in soil and naturally found in water, which makes sense in this particular order, right? So these Pseudomonas in itself is found naturally in the soil anyway. All right, here's a fun one. Okay, so Legionelles, this is an order of gram-negative bacteria. So we typically can grow these on artificial media. And so when looking at Legionella in itself, have you ever heard of the American Legion group? You guys heard those before? Do y'all have like a Legion Halls out here? Do y'all have that? You have Legion Halls? Like, I know back home, our Legion Halls, they play bingo there. Like, that's what my grandma goes, I go with her on like Fridays when I'm back home. They play bingo at the American Legion Halls because they're very nice. The purpose of that is for our veterans. So back in, I forgot the year, I think it was like 1970 something in Philadelphia. They were at a conference for these veterans. This is before then because only men were able to go. Women weren't able to serve in the military then. So it was only men at this conference, and these men were very ill. And unfortunately, about 11 men died after this conference. They went home from this Philadelphia conference. And so epidemiologists, of course, did their work, tracked it all back to the conference. And they were able to say that there was a infected water tower, a cooling tower specifically, at the hotel at which they stayed at that was contaminated or infected with Legionella. Yeah. So these men unfortunately contracted Legionella from that particular bacterium that was passed through passed through the hotel vents because of the air conditioning setup, right? So if you guys, like, I would always pay attention to that when you visit hotels and stuff. Like, for instance, back in 2019, I was in Atlanta, Georgia. I was in Atlanta when they were like on the news, there's a Legionella outbreak at the Sheraton. Where? Because I'm not staying there. I'm in Atlanta right now. So that was in 2019 summer. Basically what happens is they have to go and clean the cooling towers, the heating towers of the air conditioner units, and just there. That's why HVAC is a very important job. Like, I know they charge y'all an arm and a leg to come out there and take a look at your air conditioning units. It's because there's a lot of processes with that. I know they charge you what, $200-300 just to come look at it. Oh, they do. Just giving you that heads up. When you get a house, it's about $200-300 to look at it.
So was it named after
Yes,
the event, or did it already have the name?
No, so the name, they found it out, they discovered it right then. This is where it was founded. It was discovered at that point. So they discovered it at that point and they named it for the event, the Legionella. So it's called Legionella pneumophilia because it's going to affect your respiratory system. So it was named for those gentlemen, unfortunately, that were that passed on from that infection. They were older gentlemen, of course, because they had served in the military already and they were coming back for like this little event. So yeah.
Oh, so sorry.
Yeah, sorry. Yeah, ready to parade. So it's, I mean, to me, the story is, I like it, but I mean, it's a bad story to tell, it's an interesting story to tell, right? So it causes Legionellosis, found in streams, warm water pipes, and cooling towers. All right, so Coxiella. So Coxiella burnetii, it causes Q fever, which is transmitted via aerosols or through milk. And so what's interesting about this one, so the Legionnaire's disease outbreak was in 1976. So I was right, it was 1976. I said 77, but it was 76. So when talking about Coxiella though, Coxiella is interesting because it contains a spore-like structure. It's not a spore, okay? It's not a spore, it's a spore-like structure though. All right, so it's not a spore, it's a spore-like structure. And that's really interesting for Coxiella burnetii, which causes the Q fever. And this one is typically spread by farm animals. All right, this is typically spread by farm animals, so like your sheep, goats, cows, or cattle, should I say it's a better name? Sheep, goats, cattle, things of that nature. All right.
So, for that spore-like structure, I mean, how does this help the bacteria?
So,
Or how is that?
so with that, with the bacterium, that if it has the structure that's spore-like, it can allow it to kind of thrive in instances where it shouldn't, but it's not a spore though. It's not a spore. All right, so these guys can cause some serious issues when it comes to antibiotics, because you have to have, so the issue is that they have to have long-term treatment for this particular one. So the case can be very chronic, and long-term treatment have to take place for this particular infection. All right. And so when talking about it, unfortunately, humans can pick it up through the aerosol. So typically it's by animals. So if it's starting off with animals, and then we can get it from that, what's that called? Zoonosis, zoonotic infections. Yep, good job. All right. So when looking at this particular example here, it's going to show that endospore-like structure that I was just telling you guys about. So here you have this little cell, this blue cell that's already divided, and the little orange portion of it is like the endospore portion, and it makes the organism able to resist heating and being dried out, right? So that sounds like an endospore, but it's not an endospore, right? Sounds like an endospore, but it's not an endospore. We know that within our endospores, they look like that too. Like if I have a rod-shaped bacterium, the endospore is typically on the inside, either on either end or in the middle, but it's somewhere in the side of that cell, right? Yes.
I still kind of don't understand that, So can you kill the rest of the bacteria and then like the endospores there, and then
So,
it'll grow back from that, or?
That's
I
that's a
don't-
very
I don't get what's going on.
Yeah, that's a very, very interesting thing. So when talking about this particular bacteria, what ends up happening is that if you were to affect the other portions of that particular cell, so there could be instances where the endospores are freed because the cell is killed. That can happen. That can very much happen. But for most cases, what can also happen is that the endospores present in itself can deter these other infectious agents from destroying that particular bacterium altogether. Right, so for the most part, you have this endospore that's allowing it to thrive because this cell can like dry out and not be affected by the dryness of that environment or the lack of moisture in that environment or that particular exposure to certain chemicals. Like the bacterium is going to be unaffected by certain chemicals because of that endospore being present and it not allowing that structure in itself to be completely dissolved or destroyed. And so the endospores, if they are released, the problem is that they can then go on to ultimately cause for the infections to not be treated fully because you still have remnants of that bacterium,
And that part's
courtesy of that.
toxic.
That part is toxic in the sense of you can't kill it
But it seems like
unless
you should be able to cut the rest of the bacteria.
see
because-
out around it.
But that thing makes it somehow just be resistant? to
Yes.
stuff?
Yep. So like for instance,
That's
let's think about anthrax. Alright, so let's think about anthrax. So with anthrax is very difficult to kill. Alright, and that's because of that endospore being present. But the anthrax in itself though has its own structures that are very difficult to be targeted anyways. By being a bacillus is a gram-positive organism, so it has that very thick peptidoglycan layer. It has its own enzymes that are readily allowing for the process to take place. Its own nucleic acid, right? And so there has to be targeting of these different sites. But if the endospores are present and allowing these cells conditions to be unfavorable for those targets to actually be able to thrive and to exist, it's not gonna happen. So the endospores allow for the cell in itself to be in conditions that those other things won't work in. So if you dry it out too much, the antimicrobials won't be able to work.
It makes a little more
And-
sense.
And the only things that will essentially work for endospores would be like your, of course, an autoclave. Autoclave definitely gonna work, but certain oxygens, like those highly radicalized oxygens, they can kill it, but that's dangerous. It's very dangerous. Like we can't even be exposed to that and be unaffected.
Mhm.
Does that make a little bit more sense?
Yeah, more.
Okay.
Thanks.
I'll get some more resources for you, because we like to keep track of these resources. I'll add some more resources on there for you I'll make it on this sticky note. So resources on how efficient endospores are. And why. So I got you on that one. Alright, so with that being said, let's check out the Vibrio nalles. So Vibrio nalles is another order of this particular grouping. So Vibrio, we've seen that before, right? Vibrio. So these are going to be aquatic environments, found in aquatic environments. So Vibrio cholera causes the gastrointestinal condition cholera, water contamination, right? So water contamination is where you typically see this. So you don't really see this in more developed countries. You see this in underdeveloped countries typically. So you also have Vibrio perihemolyticus, which is going to be causing gastroenteritis, right? So you have these different types of Vibrio, and so this one causes the gastroenteritis, all right? So when looking at this particular one, it was always something I like to tell you guys about the Vibrio. I didn't write it down for this particular example, but there's something with this one that I really wanted to tell you. So I'm gonna make sure I go to my notes real quick and pull it up for this one, because I think there's something with, it's something to do with shellfish. Because I remember this one, I'm allergic to shellfish, right? I typically pay attention to things that's associated with shellfish because I'm allergic, right? I wanna know. So with this one, it actually causes, issues with, if you eat undercooked shellfish, you can unfortunately pick up this particular condition. All right, so you have to be very, very careful with undercooked shellfish, raw shellfish, because it can make you very, very sick with this particular type of microbe. And when looking at this one, it's called I think it's PSP, paralytic shellfish poisoning. So get that right quick. Because I remember that, I just didn't write that note down. Paralytic shellfish poisoning. Yep. So you can get PSP from this bad boy. Paralytic shellfish poisoning. PSP. Paralytic shellfish poisoning. You can pick up that one, unfortunately. So PSP, paralytic shellfish poisoning. Paralytic shellfish poisoning. PSP. Alright. From Vibrio parahemolyticus. Right, so something else to remember. Alright, so this is showing us a picture of Vibrio. Vibrio is comma shape, right? So whenever you see Vibrio, that shape is gonna be a comma-like shape. So what else do they have? It's a comma shape, but we see something else. Flagellum, right? There's a flagellum present, right? So they're motile, right? So these guys are motile, they can move, and it makes sense because they're in the water, right? Alright, so now we're into the ones that we should know about these. The Enterobacteriales, that's an order, right? Of what? Enterobacterius, enterics, right? Because we know the enteron, that particular prefix tells us something about the gut, gastrointestinal, right? Something's going on here. All right, so these are commonly called the enterics because they inhabit the intestinal tract. Now, this is some key characteristics about these guys. I told you this in, remember I told you guys in lab that those characteristic classifications, I was like, they're oxidase negative, right? They're gonna ferment the carbohydrates, right? These guys are facultative anaerobes. They'll grow in the presence of oxygen. They're gonna grow very well in the presence of oxygen, but they prefer to grow in the absence of oxygen. But they'll grow. They'll grow in the presence of oxygen as well. They're peritrichous flagella. What does that mean? Flagella all over. All right, so peritrichous means that the flagellum. So this one is, that's the polar. This is peritrichous flagella right here. This is everywhere. This is. All right. All right, so let's jump into them. All right, so I do want you guys to know these. Y'all gonna be so mad with me because this, for the gamma-protein bacteria, like everything. But no, in seriousness, I'm gonna go through and tell you which one's the gamma-protein bacteria. It's a lot, but just bear with me. I'll tell you guys which one. All right, so when we're looking at these guys, this particular group, E. coli, we should know E. coli. E. coli is the most commonly known enteric. Right, it's our model organism. We know a lot about it. We know its whole genome. We know a whole lot about E. coli. It's also an indicator of fecal contamination. What does that mean? Something about stool, right? Stool to mouth contamination. Absolutely. So there's this app, right? There's different apps. I have this app on my phone called the Punch Train Conservancy. This is the one that we have back home. I don't know what it is yet out here. Gotta figure it out. Still working on that. But this particular one tells us about water quality. And so, being from Louisiana, water's everywhere, right? And these jokers will get in water at any time of the day. They don't care. It could be raining, flooding, so they will get in the water. So we need to know what the water quality is like because you just don't get in water's body and not know whether or not there's contamination going on. But they do it anyway, they don't care. So with the water, they will tell you whether or not there's fecal contamination in the water. So they'll tell you whether or not there's coliforms present, the enterococci that's going to be present as a way for us to like, this one failed because there's going to be fecal contamination going on. Enterococci is present in high enough levels to where they think that you could potentially get sick. And so they do this every week. They check the water levels. They check the possible fecal contamination indicators for that. So E. coli is an indicator of fecal contamination. Some strains can cause foodborne disease and urinary tract infections, right? So that's why it's very important when using the bathroom for women, you wipe properly from the front to back, because if not, if you wipe from back to front, you can pick up that bacteria and you'll not have a UTI. All right, so make sure you guys remember those things about E. coli. All right, very, very important one, very common. We actually have E. coli normally in our guts anyway, right? Normally found in our guts anyway, it's non-pathogenic. So we know salmonella, right? I would hate to get salmonella. My goodness, it's horrible. So salmonella we know is pathogenic. It can contaminate food. And salmonella has 2,500 cereal bars. What's that? Cereal bars. We mentioned this before. The full word, serological varieties. Remember that? I don't remember.
Ones with different antibody-antigen interactions.
Yeah, so when talking of that's true. Okay. So serological varieties, this means that it has different types or versions of this particular bacterium, right? So serological varieties means that you have different types of that particular bacterium. Its immune responses can be different as well, right? Because of the serum, how the serum concentration can be, or how it can impact the serum concentration. But nonetheless, though, we look at how we have these different types of salmonella. right? You have different varieties, different types of salmonella. And so it's a very common form of gastrointestinal illnesses or foodborne illnesses. And we have salmonella typhi, which causes typhoid fever, right? Which can lead to like, very acute fever, chills. You can hemorrhage unfortunately with that particular one if it's too crazy. But that's not common though.
Okay,
Very
so
uncommon.